And so we're going to build structures on all amino acid chains, then restrict them to things like proteins. And the proteins, there's a protein database. It's a wonderful thing on the internet. It has listed pretty complete descriptions of about 100,000 proteins. So that's the main proteins on that list of 100,000, and about a third of those are synthetic, and I don't hardly count those. They're not found in people or in animals or in nature. And some of them are almost the same, are so similar to each other that they hardly count as separate proteins. So that's not a terrible number, not a terribly big number. Things I talk today will be proteins or subsets of proteins. So everything is going to be done today in the framework of these clean objects, these neat objects. So it's extremely mathematical. And later on, we'll look at these in more detail, because I think this problem I mentioned, the function and structure of proteins goes beyond the amino acid chain. To go deeper, and this is, I think, the big problem of biology, is how to understand the higher structures here. This is called sometimes the primary structure of the protein, or the linear structure, and it folds. So the protein, you can think of as a long, wobbly string, and eventually, in fact, soon after it comes into existence, it starts doubling back, folding across itself into a three-dimensional object. And then it's an object in three-dimensional space that has a certain three-dimensional geometry, and a big problem is to understand that geometry in terms of the sequence. And it's the main dogma, which I'll make a big deal of, is that this sequence determines the whole function and structure of the folded object. The three-dimensional protein is determined by this. That's called Anfinson's dogma. And it's sort of central to my way of thinking about these things. Now, I'm getting a little ahead of myself, but I want to emphasize that these are such important objects for biology, and they're things that we can use a lot of mathematics on and do. Okay, a peptide, biologically defined, is a fragment of a protein. That's the definition. So it can be expressed as a small amino acid chain. A fragment could be expressed by all the chains are in length, typically for purposes of this peptide binding problem, around 15, length of 15. And they are, historically, they're fragments of proteins, so they come from living bodies, but will also allow the possibility of synthetic vaccines too. So we can go beyond just the ones that are defined in the strict way as fragments of a protein. We go beyond that to look just at general sequences, kind of special sequences of amino acids. Okay, so we want to look at the peptide binding problem. So we want to talk about binding to these molecules to an allele. Allele also has a representation as a sequence of amino acids. So everything is in the same domain. An allele, pretty precisely, is a reflection of a gene or a variant of a gene in an individual. From simple genetics, even I used to know a little bit about this, that a gene has a different form in different people. And that's the allele. Same gene is a class of alleles. The different alleles are representations of the gene in a person. There aren't so many. We worked with alleles. So these are going to be, let me just say what this space is. There's about 800 alleles. And I'm going to, this word will probably throw you. It bothered me for a long time. I still don't know if I could say it. MHC2. This is the major histocompatibility complex. And that's the set of these genes or alleles that are associated to the immune system. There's two classes, MHC1, MHC2. So we started out our whole project working with these alleles, MHC2. Those are the ones that are involved in the immune system of the humans. So we're going to be looking at these alleles. So the binding problem is connected. A set of the peptides is going to be bound to a particular allele, one of these 800. So that's the setting. The alleles have length. Well, we work a lot with the databases, and so we've taken out the main section of each allele to get them all of length 81. So these are 81 length. This is kind of a normalization. 81 length amino acid chain. So this is a protein. These alleles should be thought of as proteins. Okay? So this is kind of a setting. We're getting into the mathematics more and more as we proceed because we're working with all these objects which are the same type. So what we want to do is to look very broadly. Let's take all possible sequences of amino acids of all lengths, arbitrary. And then we restrict a kernel we obtain on that to peptides, or we restrict it to the alleles, or study that on proteins. So that's the idea. We can define things on this huge universe. It's forbidding how many amino acid chains there are. In general, you just take, n to the 20th power, something like that, or 20 to the nth power. It's a huge number, though. Big combinatorial number. But we still go ahead and define a kernel on these. Okay, so here's this construction of a kernel, which is very similar to a kernel, as I mentioned yesterday, for Google problems or for vision problems. Similar kernel. And this is going to be a three-step kernel. So K1 is a kernel chains of length 1. So it's a kernel on A. So we start with a kernel on A. And this is where the biology enters, or the medical information biology enters on the kernel K1. After that, everything is automatic. It's all mathematical. But this kernel here, it can be represented by a 20 by 20 matrix. It's called the Similarity Matrix, because the idea, if the values of the kernel are large, it means that the pair are similar to each other. The larger, the closer they are. If you normalize, then you get 1 is the largest value, and then the pair in the kernel, if it gets close to 1, means very similar, and each one amino acid is similar to itself, so K of AA equals 1. So, well, I could spend a lot of time on this. We spend a lot of time on what's the best choice for K1. And biologists know very well, they don't use the same language that I do, they never worry about positive definite, which for us is a crucial thing. But they understand the importance of the similarity. They're not talking K1, but they're talking about similarity, or they say substitution matrices or mutation matrices. And that's a very big thing in biology because these 20 amino acids are basic for life. So to have this idea of how similar is one to another, close to the idea of how likely it's going to mutate to the other one. That's why they sometimes say mutation matrices or substitution matrices. There are well over 100 suggested kernels in the literature. Well over 100. And we spent some time trying to find which would be the best one for us. And the most common and probably the best one is Blossom 62. And I won't try to define it here, but what one does is it's a mind-boggling process. It goes from the data of all proteins. You look at all the big database of proteins called SCOPE, S-C-O-P. Then you align them into blocks with similar function. And in those blocks, you align them so that the positions correspond roughly. And then you do a big counting. And after all this is done, you get this blossom. And then you get the Blossom 62. Okay, so it's very famous. Just about everybody in biology, at least this part of biology, knows very well Blossom 62 because it's the main substitution matrix. Okay, so it's a little horrifying when we read this. The last stages, what they do is they have this matrix, could be interpreted in terms of probabilities, etc. And then numbers are maybe a little big. They take the log. Okay, they never worry about whether the matrix is positive, definite, or not at any stage. We proved it was. But considering it's such a main object, people have never thought about whether it is a positive, definite because biologists never saw the role of positive, definite. Okay, so what they do then is take the log. Now it's certainly not positive, definite. It's not positive. It starts as a positive matrix, take the log, and it's a matrix then. And what they do next is to round off to the nearest integer. And these integers are usually less than 10. So it's a huge destruction of a property to round off to these integers from 1 to 10. That's a little horrifying for us, so we don't do any of that kind of round off. And we do something else. We take the precursor of this matrix, which is hard to find in the literature, the precursor of that, then we do some normalizations, and eventually we get a modification of Blossom 62. We just write -2. It means our modification of Blossom 62, and that is going to be K1. Well, there's a parameter here. So this is a matrix, symmetric matrix, positive, definite, and we can take the power, Hadamard power to the beta. So let's do that. So beta is a parameter which we don't really understand. It might be same beta that's used in dynamics and works with people like Ruel and Bowen for this thermodynamic formalism. They have a beta that might be very much the same beta here. I don't know. So anyway, the beta is a parameter. Naively, you could take 1, but we usually end up beta equals 0.1, 0.06, depending on the space we're looking at. So that's what we choose, and we choose that by a lot of numerical experimentation. We get such a number, a power. This is a Hadamard power. It's not the power of the matrix. It's the power of the elements of the matrix. So you take each element of the matrix, take it to the beta power. So let me emphasize this is Hadamard. And one thing it does accomplish that we know is eventually we're going to be taking powers of K1, very high powers. And if in these very high powers, you certainly get much too big numbers. We might have a 50th power in our construction of the next stage, K2, and this beta makes it more tractable, because take something with 50th power and beta is small, like 0.06, and it's not such a big number. Okay, anyway, that's part of this analysis of the first stage. So, as I say, it's constructed with a lot of biological information in the Blossom 62. It has to do a lot with the functions of these proteins in this big database of proteins where all the amino acid chains are laid out, and then they line them up and do these counting arguments depending on the function. So it's very highly oriented to the biology, the function, and structures of these things. Medical evidence and things like that. Studies of proteins. Yeah?
Small question. By definition, the elements of the matrix should be positive.
Should be what?
Positive. The elements.
Yeah, this is positive. Blossom 62-2 is positive. See, we're taking the pre-Blossom 62 in this construction. The pre-Blossom, Blossom 62 uses a positive definite matrix, and then they take the log of that. So the typical Blossom 62 is not positive elements. Ours is. We've used other matrices too, but mostly we use this for our first stage kernel. This is then a kernel, positive definite matrix, defined on the space 20 elements A. Function on the product, A cross A. So it's a 20 by 20 matrix of positive elements. Okay, so that's the starting point of our construction. Okay, now we'll say K2, this is pretty easy. K2 of UV. Now we take U equals U1 up to UK. These are going to be amino acid chains. V equals V1 up to VK, same length. And here we just write product I equals 1 to K, K1 of UI, VI. So this extends just by, it's a tensor product or product construction. Very simple mathematical construction, which goes from chains of length 1 to chains of length K for any K. Any K. And these are called K-mers in biology. That's a K-mer. So it's defined on a pair of K-mers, this kernel K2. Okay? And now what one wants to do is you go to K3. That's going to be kind of a, it'll be a similarity. You could call, it's inspired a lot by our vision work, but it's also inspired by local alignment, what's called local alignment kernels. So we can now take K3 to be defined on FG. And all things are finite here. All these sets are finite. So amino acid chains can be infinite, but we're always going to be talking about finite subsets of amino acid chains. So keeping everything finite. So F and G are amino acid chains. And this is going to be by definition. By the way, there is a little bit of work to show these are positive definite because of this power. But it is. We prove that. So this is going to be now the sum, or you could take an average here, of K2 of UV over all U contained in F, V contained in G, and K. I could put a K, sub little k here, but this works for each K. And U contained in F means a connected substring of fixed K for each K. So this is going to be a K-length substring, continuous. So we have F1 up to Fn. Then we take U here will be of the form Fi plus 1, Fi plus 2, up to Fi plus K. Okay, so it's a continuous substring of F. And the same for V and G. And we're taking the sum over all possible pairs. And each of these pairs, we have this similarity. All these are similarity matrices, can be thought of as similarity matrices. And so the similarities are on here. And we want to measure the similarity between these two amino acid chains in terms of all possible pairings of the substrings. So we're looking here, for example, U in here. G might be G1, and this could be G different length, and this could be over here. Two substrings of length K in very different positions in the two amino acid chains, F and G. So we're trying to sum over all possible pairs. Because, there's a big motivation from genetics here and similarity, because I can't really go into that, not so well anyway. But this is based on a lot of work in genetic biology, having to do with what's called local alignment. There's something, maybe I'll get to that later. So this defines our object, our kernel. This is the
biological meaning of this K2?
K2?
Yeah.
Oh, it just extends the initial K1 to these strings of, this just extends it in a simple way to a strings of length K. This is just a product, so this is just naive extension. This is a product. Yeah, so here you have U1 up to UK, V1 up to VK. So it just takes these two paired and looks at their K1 similarity. Then you look at the same thing for the second and so on up to K, multiply them all together. So it's a very naive and simple extension of the K1 similarity to strings. Here is when it gets more subtle and more complicated, because here we're looking over all possible subpairs and their similarity. Yeah, so this is a complicated object.
But biologically, they don't mean, what? Yeah, I was wondering what kind of biology is behind that.
Oh, all based on the amino acid similarity, yeah. See, all the biology is in the amino acids. So once everything is based, it's all mathematics after that. Yeah, so you can't easily say the biology here. The biology is already finished with K1. Questions on this? So this gives the construction that I've been talking about. This is a construction of our basic kernel. And we can write, as usual, K hat 3 of F and G. We just have K3 FG divided by K3 FF, K3 of GG square root. This makes everything's positive, so this puts things in the positive ortho, and it's going to look something like this in high dimensional space. Everything is going to be around in here. So it's this region right here in the high dimensional space. It doesn't get too small. It doesn't get too big. Never gets bigger than 1. Okay, so this is the kernel we actually use. And so K3 hat is very nicely behaved because of this normalization, as far as not getting too big, too small. Never gets anywhere near close to 0 or never gets bigger than 1. And these matrices, the diagonal is always the biggest elements, 1. The diagonal elements are always 1, and everything else is less than 1 and never gets too small. All positive.
What happens if you take another K1 stuff?
Oh, well, that's a good question. You can, we do that, and that's a question I'm still intrigued about. Other ways of looking at K1. It's a big subject. Choosing what does it mean to choose K1, and how to do it best, and so on. We have another one we tried that maybe I'll say a bit later on completely different principles, and this is just a few weeks ago. We tried that, and it was good. Not quite as good as this. And we measure things in terms of the peptide binding problem. So we have, that's one thing, we can measure the theory a lot by using the data in the peptide binding problem, see how well the theory reproduces that data. So the data is a good test on the development of the theory. And that's a trademark of what we do. What we do is a hugely computational. Computations take days, weeks, oftentimes. And the computations are guided a lot by the theory at a certain point. The computations come back, we adjust the theory. And so there's all this feedback going on between the computations and the theory. But it's a very big computational project because, the main thing is this kernel is, if you have a database like, how many here would I say, 800 of these elements of the gene database, so it's going to be an 800 by 800 matrix. And each of these elements in the matrix has got huge sum over all pairs of subsequences. So you can see there's a lot of fancy programming tricks needed to even make this feasible, computing this object. But in the subject we're in, we don't need fast computation. Fast computation, you have to be fast enough so you can actually do it in your lifetime or in weeks. But on the other hand, it's kind of a research where very speedy things are not important. If you want to do speedy things, you can do that too by taking random subsets here. You can do a lot of things to speed it up. But every time we try to speed it up, we lose accuracy. So we want for us, accuracy is more important than speed.
When you say looks good, what do you exactly mean by that?
Oh, it looks good in the terms of eventually the algorithms which predict protein binding. So because there we have a lot of, quite a bit of data, and we can see, we know from those databases whether our structures are working. We need to. What I often say is, are we doing things sanely? What we're doing is there a sanity part of what we're doing? And for that, we always do a sanity check by looking at the data and seeing what comes out. And, then we can see something wrong pretty fast. It'll turn up. Okay, so we have this kernel, and how do we use that? So this is an algorithm which I spent a long, long time making error estimates for this algorithm called regularized least squares. I'm not listing a lot of names. And our paper that I mentioned, you can find on Google and so on. You can find a lot of details, including references and so on. But this is the algorithm that we use. In this algorithm, I did a lot of work 10 years ago with Felipe Kucher and Dingshuan Zhou on the mathematical foundations of learning theory. And that all was revolving around this algorithm. That goes as follows. So we take a space X. So in this case, X would be a space of peptides. Okay, so here X equals a peptide space. So the number of peptides we study is going to be somewhere around 100, 500, sometimes 1,000. Okay, and here we want to write a kernel, K sub X, on this space. These are all peptides, and actually they don't fold because they're so short, so there's no three-dimensional aspect. So the linear picture here is pretty good. And so we take K3 that I described right over here. We take a hat version on that. So this is a kernel on that space X, okay? And the kernel represents, in a strong sense, biological reality. Okay, so then what we'll do is we'll look at the following minimization problem. We minimize this function. We have data of the form say xi, yi. Data is a form like this, I going from 1 to m. So xi belongs to the peptide space, and yi it just goes between 0 and 1. Okay, so that's the data. So we're looking at the data of a function, a peptide binding function on X. So we're looking for this function f going from X to 0, 1. We want to estimate this. This is going to give us the degree of the binding. Another thing we differ a lot with other people working in this field, we emphasize the binding, the degree of the binding, not the question, does it bind or not bind? Just about everybody else looks at the question, does it bind or not bind? But binding is a matter of degree. It's an arbitrary, a little arbitrary line. And so the convention we use in binds, P binds to an allele A. So we have a fixed allele in the background. So you have a fixed allele. So we're looking at the problem of binding a peptide to that allele. That's the one that starts the immune process. If P binds to A, then that's the main stage of that allele to signal to the killer cells to release, to kill the invading virus. So P binds to A is going to be given as a function, and it's a threshold if f of P is the same as x. P equals the x over here, xi. F of P, a peptide, is greater than 0.42 approximately. There's a number there. So this is a threshold for binding. So what we emphasize is this binding affinity as a function, and you can always recover the usual binding, not binding dichotomy. But there's nothing that special about the threshold, so if you have f of P close to it from above or close to it from below, it's the same binding qualities. And binding has to do with how close the molecules are. So it certainly is a matter of a degree here, not yes or no. Okay, so that's the function we get. And it's going to be the solution of this problem. We want to minimize sum of xi. There's a certain plausibility to this over I equals 1 to m, the data. We want to minimize f plus lambda norm of f squared in this H Hilbert space, or in this norm defined by, in the Hilbert space, by k hat 3 on x. That's the equation that has to be satisfied. So this is naturally, minimizing this is going to put f into, well, these are finite spaces, so it gives it, but it will have to have a finite norm in that space. Of course, everything's finite. So most of this theory is infinite spaces, but here we apply it to the finite spaces. And so this is our problem. Minimize this. For a given lambda. And then you can, just a little bit of work will give us the solution. It's a linear algebra problem, solving a linear system of equations of order m from this minimization. Because everything's quadratic here, so it's very simple to write down a specific algorithm which gives that solution. So it's a very simple algorithm, robust. And this lambda is an important parameter. See, if lambda were 0, then one would get something like interpolation, which is a very, very poor algorithm. And so lambda takes into account noise. Lambda takes into account noise. So that's crucial here. So this is classic, I call classic regularized least squares. Goes back almost to Gauss. It goes back to Tikhonov in Russia 50 or 70 years ago. And it's I think, a central algorithm of mathematics. And now one chooses lambda. And this is done by standard algorithms where you try one value of lambda, and you test. So you test on part of the data, and then you train this on part of the data and test it on the other part of the data to be able to get a good lambda. You get good lambdas that way. Cross-validation is called. So basic part of the algorithmics of this kind of approximation theory. So here's approximation theory. Okay, so that has a solution. We call that solution f sub lambda. That f sub lambda is, in fact, gives us an f here. I've condensed things a little bit, but I've given a pretty complete sketch of the algorithm and the mathematics here. This is a pretty complete picture. I mean, I've rushed over a couple of things, but that's pretty complete. And so this, for a fixed allele, we get this we call f sub a, depending on a. And then we have a set of peptides, as I say, this big, which you might say is associated to a, because the people who have done the medicine think that this is going to be a natural set of peptides which could bind to that allele. Okay, any questions on that? Yeah.
How do you know how to choose this lambda?
Oh, because to choose a lambda, you do this cross-validation, which means you take the data, say split it, you can leave one out and test it, get a function here which doesn't depend on the one you left out. And then the one you left out, you test the function you got. And then you do that, repeat that, and that gives you a better and better lambda. So you can solve for lambda. Because you can use part of the data to get a known prediction, to get a prediction on something that you know already. And that will give you a way of narrowing down the lambda. Okay? Maybe not too clear. See, so if you let's take this problem, and let's just do it over, say, the first n-1. And that will give us a certain function, which depends on our fixed lambda, and then we can see if the solution there, which does not involve the nth data point, we can test it on the nth data point. And if it's a good prediction, that means this lambda is better. And you keep doing this and doing this and doing this, and finally you get a pretty good lambda. You do this by leaving one out over all the possible m's. So that gives you a way of honing in on the good lambda.
Well, following Tikhonov, there is a whole school of regularization theory. People have techniques on finding lambda and so on.
Oh yeah, cross-validation goes back especially to Grace Waba.
Yeah.
And there's another cross-validation here too when we compare. There's another thing called five-fold cross-validation, which we use a lot too. Most of that's in our paper. So you can read that. Okay, so at this point, we want to say what we have here, how does it look? Okay, so there is, I mentioned, a lot of people doing this, looking at prediction methods for this peptide binding problem. And so there's one lab, one group of people, in Denmark, Nielsen is he, and Lund is another guy in the leadership of this lab. This is Nielsen, Morten Nielsen. And they use a nonlinear neural network algorithm, something that's immensely complicated with immense amount of data going into the algorithm. So they had the best results, better than everybody else. They're always doing the best. So he always says he is the state of the art. And he does. So what we always use his results to aim for. We always say, how are we doing relative to Nielsen? Because this was so good and so much better than all the other people. But the algorithms, you couldn't learn much about it because it was such a complicated procedure. This nonlinear neural network algorithm is complicated. Okay, so when we do this as a standard dozen or so alleles, then we average over the alleles like everybody does, and we get the results in our paper, which are, so we do better. And so we have better than Nielsen. So when I first got in communication with Nielsen and sent him some of our stuff, he wouldn't believe it. He said it can't be because we didn't use anything much. And so it took a while. We had some more correspondence. He worried that we're had some kind of bias in our method, which is unfair bias, overfitting. So I convinced him we did everything straight. We eventually used even his data, and not only that, we used his randomness. We used the same random sample, so it wasn't randomness getting in the way. So we used the same random sample, came out always better. And so more recently, Nielsen says, sent me another mail. He said, we've checked, we put the algorithm and everything so they can follow it. Everybody can follow it. He said, we've checked everything in their lab, and indeed, we do as great as I said. So he said, in fact, great thing that we did, because we're using so little here, in contrast to what everybody else was using much more high-powered ingredients in their algorithms. And another thing that's important here, I mean, there's a huge story that I'm not getting to, or I don't expect to, but one thing that we've used is noteworthy. There's something in the foundation of all this whole study of proteins. It's called alignment. So alignment is the universal and foundational for the whole study of this genetic bioinformatics. The idea is you take the proteins or protein-like objects and you align them up so that they match as well as possible, and you have to modify them a little bit. You delete some and add some ones in the sequences to get everything aligned together. So if you have a database of even 600 or 800 here, you align them all up so that now you make sense to talk about the 15th position. It'll be the same for all of them. And this is really a big part of the foundations of this whole work in bioinformatics. And so what we do here, we don't use that. We avoid alignment. What we do, indirectly, alignment comes into that matrix BLOSSOM 62-2, because there's a little stage in that process where they use an alignment. So it's not quite fair to say in our paper that we don't use alignment, because we use BLOSSOM 62, where it's just in the first stage there. But since then, we have another amino acid substitution matrix, which is derived without using alignment, and we get results with that just a little bit worse than our first results, but still better than Nielsen. So we get better than Nielsen without using any alignment at all. And so that's, I don't think our stuff is well known enough, and we didn't say it so clearly enough in our paper, because we still use a little bit of the alignment. But now we have eliminated alignment from this project. So anyway, that gives our first main result. We got, I don't know how long ago. This quite a while. Maybe it's over a year ago, and we've been developing on that. And so yeah, but this was the first main thing we got. And Nielsen now is sending his main student, finishing up a PhD with him, to become a postdoc in our lab. So Nielsen's a very good student who I met. He's coming to work for us as a postdoc. Anyway, that's the picture. Thanks very much.